Hey guys! Today we're going to be talking about protein synthesis, which is one of the major concepts in biology, and it's the basis of all genetics, and it's super cool. So I hope you like it. So we're going to start with RNA, right? We've already learned about DNA. RNA is very similar, except it's single-stranded, right? DNA has a double-stranded helix, right? Like this little picture on the side. RNA, or ribonucleic acid, is single-stranded chain of nucleotides. It has the same bases, except instead of having a T, it has a U. So A, U, C, and G instead of A, T, C, and G. Okay, so here's a look just comparing RNA and DNA, right? DNA is our double-stranded helix. RNA is the single-stranded. They both have cytosine, guanine, adenine for nitrogenous bases, but where DNA has thymine, RNA has a nucleotide called uracil. So that's another difference. All right, so what is a gene? Because RNA is a copy of just one gene from the DNA, so we need to understand what a gene is. So a gene is just a segment of DNA that codes for a specific protein, right? All of our DNA codes for all of the proteins that make us look and function the way that we do. So each segment of DNA that codes for a specific protein is a gene, right? So if we're looking at this little chromosome here, this pink segment is representing one gene. So for example, we have a gene for keratin, which is a protein that makes up our hair, right? People have different colored hair, different textured hair. That all depends on the amino acid sequences, the nucleic acid sequences in that gene. It's about 470 amino acids long, and it's an example of a gene. All right, now let's get to the good stuff. Protein synthesis. The word synthesis means to make. So we are making proteins. This is the basis of all biology, the basis of all genetics. It all comes down to making proteins, which is coming from the instructions from the genetic code. So we're going to see the flow of information from DNA, which we've learned about, RNA, and into proteins. So three major things are happening. We've already learned about DNA replication, right? When DNA makes a copy of itself, a full copy of the entire genome that's in your cell. We've learned how that happens already. Today, we're going to focus on transcription and translation. So transcription is just when we take one segment, one gene, and copy it into an RNA strand, right? Remember that single-stranded nucleic acid message. It's just one gene. We're not copying the whole, all of the chromosomes like we did for DNA replication. We're only copying one gene this time. Then we bring that to the ribosome, and the ribosome translates this from a DNA language to a protein language, amino acids. And that's how we make a protein. Okay, so we're going from DNA to RNA to protein. And the protein is what makes up our traits. So let's see if you can label these. So we've got something here, transcription happening, turning it into something else, translation, turning it into something else, which is the traits that we see. So we've got DNA making a copy into RNA, turning that into proteins, which represents our traits. In this case, we've got some glow-in-the-dark mice, which is something you wouldn't see in nature, but we can make in a lab. We can make everything. Okay, so let's see more specifically how this happens. Starting with the process called transcription, right? If we go back here, that's going from DNA to RNA, really simple. Okay, so we're going from DNA is getting copied into RNA. Only one gene. We're not copying all the chromosomes again. We're copying one gene into an RNA message. Because DNA is really important to the cell, it doesn't leave the nucleus. But the ribosomes are the guys that are making the proteins. So in order to get the DNA message to the ribosome guys, we have to copy it into an RNA, which is what's happening here. So we copy a segment of the DNA into RNA, which can leave the nucleus, travel to the cytoplasm where the ribosome is, so that the ribosome can use those instructions to make a protein. Okay, so first thing that happens, one gene is transcribed from DNA into mRNA. The M just stands for messenger RNA. It's like a messenger in the nucleus. Second step, RNA leaves the nucleus, carries the message to a ribosome. Here's another picture of that happening here. So the DNA kind of unwinds, just like it does with DNA replication, right? This is RNA polymerase. It's just an enzyme that does this, just like DNA polymerase is the enzyme that makes the new DNA strand for DNA replication. RNA polymerase is the transcription version of that. So this blue strand here is the RNA. So this is one gene, and it is transported to the ribosome. That's all for transcription. And if you think of the word transcription, it's like transcribe. If you're going to transcribe something, you're copying it, right? So we're just copying the DNA message into a form that can leave the nucleus. That's all that's happening here. Okay, so when this happens, right, I told you RNA has the same basis as DNA except U instead of a T. So when the sequence gets copied into RNA, right, so this is DNA. If this is DNA, let's think about what the RNA will be. All right, so if A pairs with, not put this right in the middle. If A pairs with T normally for the DNA, RNA, it's going to pair with a U. Okay, so A pairs with U. T still pairs with A. Everything else is this. Oh, I'm sorry. U. A. Again, T pairs with A. C pairs with G. Okay, now you take a second and see if you can figure out the rest. Okay, so G pairs with C. I don't know why my things are not matching up here. Maybe I have to do two of them. So what did I do here? Okay, G, C, C, G. A pairs with U. A pairs with U. C pairs with G. Hope you got that right. All right, that is all for transcription. So that's the first part of protein synthesis. Really simple. Second part is translation. This is where it gets a little more complex. So now we are translating that message from RNA language, right, nucleotide language, the A's, T's, C's, and G's, to protein language, which is amino acids. So just like we're translating something from English to Spanish, we need to go from one language to another. So it's a little trickier. So let's see how that happens. So what happens next? The RNA travels to the ribosome. Okay, so we're leaving the nucleus here. We're traveling to the ribosome, which lives in the cytoplasm. Then tRNA, which is another type of RNA, which I'll show you in a little bit, brings amino acids. Remember, those are the building blocks of proteins. So they're represented by these little circles here. So it brings amino acids to the ribosomes based on the RNA code. Okay, so whatever this code from the DNA says, that's what the RNA builds. It's like instructions for a recipe if you were making a cake or something, right? You follow the recipe step by step. That's exactly what the ribosome is doing here. It's following that RNA instructions step by step to make this exact protein. So the amino acid sequence then makes a protein when it's complete. Maybe that's keratin for your hair. Maybe it's hemoglobin for your red blood cells. Maybe it's insulin. Maybe it's an enzyme. Everything we're made out of is proteins. So we have so many different types. And all of the codes for making those proteins come from our DNA. Okay, so kind of like I was talking about. So in order for the nitrogenous bases, the order of the nitrogenous bases that carry the instructions for building polypeptides, which is a protein. Okay, so this code, the order of A's, T's, C's, and G's, or A's, U's, C's, and G's, in the case of RNA, makes a specific protein. Okay, there's something called a codon, which is a 3-nucleotide sequence that codes for a specific amino acid. There's 20 different amino acids. Each codon, so each set of 3 bases, seen here, codes for a different amino acid. Okay, let me move myself over here somewhere. So AUG codes for a different amino acid than ACG. And the way we can figure this out is by using something called an amino acid wheel. So this wheel, see, can I move this? No. This wheel, we can match the amino acid. So what you would do is you start with, here's your codon, your set of 3 bases. You start with, it would be U. I'm sorry, okay. So if the DNA is TAG, right? T pairs with A pairs with U, G pairs with C. Okay, so our codon on the RNA would be AGC. So now we use this wheel to figure out which amino acid this codes for. Okay, so we start in the middle with the first letter. So A. So now we're in the A section of this pizza pie here. Let me move myself so you can see. We're in the A section of this pizza pie. So A, and then go to the G, and then go to the C that connects. So serine is the name of the amino acid that this codon codes for. Okay? All right, now let's take a look at how this works. So remember I mentioned tRNA a few minutes ago. So this is an RNA molecule that has an anticodon at the bottom that matches up to the codon on the RNA. And it has an amino acid at the top, different amino acid depending on the codon. So that's how it knows to bring a specific amino acid to the ribosome based on the codon that it's reading on the RNA. Okay, so codons of transcribed mRNA are decoded with the tRNA molecule. So an amino acid is attached at one end, the anticodon is on the other end. And again, this anticodon matches to the RNA. Okay, so if this is our RNA strand, which the ribosome is reading, here's the tRNAs. All right, so right here, the tRNA is matching AAA on the RNA to UUU on the tRNA, which codes for a specific amino acid, lysine. Okay, same thing is happening here and here, just using that amino acid wheel. Okay, so what I want you to do for a second is give this a try. See if you can transcribe and translate this genetic code right here for hemoglobin. Okay, let's check how we did. So CAC matches with GUG. GTG matches with CAC, and GAC matches with CUG. How do we do on that part? So now we're going to figure out how. Now GUG on the code here, GUG, valine. CAC is histidine, CUG is lysine.